Cozy. We're going to start off. We have 3 talks for you in the last afternoon, 2 20-minute talks, and then our final plenary. We're going to start off with Samson Fong, who's going to be giving a talk on DiseaseScope, automated construction and interpretation of hierarchical disease models.
Thank you. Hello, everyone. I would like to thank the organizers for this opportunity to present my work. My presentation will be broken down into 3 parts. First, I'd like to convince you why we need hierarchical models in the first place. And then second, how can we build disease-specific hierarchical models? And then finally, what can we learn from it? And to my pleasant surprise, a lot of people are in this particular cozy are already talking about multi-layers, hierarchical type inference. And I would like to add to that to see why we should do this. So the main reason in our minds why we should build these hierarchical models is that biological systems themselves are organized hierarchically. At the nanometer scale, you have proteins, which then come together to form complexes, and then go all the way to the other end of to the micron scale in cells. And the problem is that traditional genomic analyses focus on clusters at a single resolution. So in this particular case, they have measured, they took more than 10 years to measure this gigantic genetic interaction network in yeast. And then they then start to cluster and then show that they can recapitulate known complexes. And we think that is fantastic, and that's a great use of the data, but we think there is more to this and that we can squeeze out more information out of it by looking at this in a hierarchical manner. So what do I mean when I say a hierarchical model? So fundamentally, it's simply a model that contains overlapping gene sets to identify biological entities at multi-scale, like protein complexes to genetic pathways. So these models would then start with small modules at this low end. Then these small modules come together to form larger modules in an overlapping fashion, all the way until you contain a module that contains every single gene or node in your network. And as a side point, we capture gene pleiotropy, or these multi-function nature of protein complexes, by allowing this hierarchical model to be a directed acyclic graph, which simply means allowing these smaller modules to come together and form multiple larger modules. So in one of our first attempts at this data-driven hierarchical modeling, we see one of its main use and benefits. So on panel A here, you have this flat network that contains a lot of physical interactions because these are all genes that then encode parts of the proteasome subcomplex. So if you measure the physical interaction, then you would expect them to be dense. But the problem with having a dense cluster like this, it's hard to make heads or tails about it. And it's not immediately clear to see how this will look like that. So when you cluster this in a hierarchical way, first you get the subclusters, which then very nicely fall into subunits onto the proteasome complex. So that's great. But then we're also able to see that these subunits actually come together and form the proteasome complex. And then if you look at the entire model itself, this complex then integrates with other things, then you can start looking at a whole cell model in a hierarchical way. So this particular work took many years, and we have ongoing works at hierarchical modeling that also has taken many years. And we believe that this is a difficult challenge, but we want to be able to contribute a pipeline that makes it fast and easy to build at least a first draft of a hierarchy so people can start to play with it immediately. And for us, we have taken the challenge of building these hierarchies in a disease context-specific way. And that effort has been born out of the NCATS Translator Consortium, which the consortium includes labs from all over the country in the US. And the consortium's goal is to generally go through the vast amount of biomedical data available to generate hypotheses. And in our participation in this consortium, we believe that hierarchies is one of the ways that we could do this to integrate data. So I hope by now that you're convinced of why we need hierarchical models, and I will briefly talk about how we can do this in an automatic fashion. So here is the pipeline for building a disease-associated hierarchical model. So the first step is to have input a disease name, and then query what are disease genes. And then you will also want to query the disease tissue if you would like to get a tissue-specific network out of it. That way, then your data source is actually more relevant to the disease at hand. And then once you have a network, you can do two cool things. One is to expand your seed disease genes, which usually is only a handful, to incorporate more genes through so you can get a richer hierarchical model. And then from there, you can infer the model. And then finally, we also have developed tools to browse and then ultimately analyze this hierarchy. So just to click through each step in that pipeline, I have an example query, asthma. Asthma is a complex and common disease, and in part, at the very least, to have some genetic basis, which makes it a perfect candidate for this sort of analysis. So the first step to define an asthma gene set, this is actually very similar to the talk that right before the break. He provided methods to go from OMIM, which is similar to this particular option, and also DisGeneNet, which is the other option. So you recover very similar information here. So for the asthma gene set, you actually get quite a few genes from DisGeneNet, which is typical. But each gene is weighted by a confidence score. So you get some interpretability, and also you can set a threshold where you want your gene set. BioThings, on the other hand, which enables this API call, for us anyway, return 99 genes, but there are no scores associated with it. So let's take a look at and see how close the two gene sets actually are. So if you rank sort the DisGeneNet, so the 1,300 genes in order, so the most confident here and then the least confident there, what you see that is the blue bar, which are genes found in BioThings. BioThings are actually finding genes that are very confident. So for us, especially for this application, we'll just stick with the 99 gene set, and then we'll use that as our seed, and then we'll carry the rest of the analysis from here. All right, so next we want to define what are the tissues affected by asthma. And the way we do this is we look at a pre-trained model, a word-to-vec model that was trained on PubMed abstracts. So it contains words that are available in the PubMed corpus. And so what we do is then compare the disease, in this case asthma, so we get a word embedding vector for that, and then as well as the word embedding vector for each of the tissue that we have in our database. And then we compare which tissue is actually the most similar based on this word embedding vector. So the similarity score, which is the cosine similarity, is what's reported here in the second column. And then the disease name on the first column. Lung being the top one, I think is no surprise, so that's actually a very positive result. Bronchial epithelium, again, these all are very positive results for asthma, so we're very happy with those. And then the percentile, what you get here is that you're comparing that similarity score to the rest of the corpus. So it gives you a sense of how specific this is. So you don't want a disease to be tissue to be highly ranked just because it's similar to everything else. So this allows you to get some confidence in this scoring method. And then next, now that you have the gene set and a tissue, then you can start to select some appropriate networks. So to us, there are some very good high coverage starting point networks that are good for starting points. So physical interaction networks, so protein-protein interaction, BioGrid is a fantastic source. If you're interested in more expression-based, so you can look at expression correlation networks. If you're studying cancer, TCGA is the best resource for that. If you're not doing anything cancer-related, though, then GTEx might be the more appropriate one. And then finally, if you just kind of want a tissue-specific network that contains some already omics integration, then you can use the giant network, which has lots of different tissue-specific networks that they have gone through their pipeline. So to get these networks, a lot of times you need to do a lot of data wrangling. So we recommend two places for you to get these networks. One, we like Index. It's an in-house general network repository. It's a great place to simply store networks as well as share networks and also pool networks. The index team has also done a fantastic job at curating a list of gold standards or very high quality publicly available networks as well. So you can visit index to get one of those. Finally, I'll also like to call out this second repository that we're calling Big Jim, which simply is a repository that contains a lot of pre-processed, pre-calculated gene-gene interaction networks. So you don't need to actually go to TCGA and download all their data and then do the correlation and do the cutoff and all of that. So you can get that and get an easy network right away. So then the next step, once you have the network, then we want to expand the gene set. So we started with 99 genes, which is a handful, but that's not quite rich enough to see to build an interesting hierarchical model. So we're going to expand it. And there are two methods for that. One, you can simply take a network and then ask what are the nodes that are next to your disease genes. That's one way to do it. The other way is to do network propagation, which then is a more sophisticated way to basically take into account the number of paths that go through the neighbors. And in using then network propagation here, we actually has fantastic recovery of a lot of genes that we added to it are actually also genes that are found in this GeneNet, which again give us some additional confidence in this method. If you're interested in finding disease genes using random walk, I would like you to check out this paper and as well as a talk by Dan Carlin, who's giving a talk tomorrow to talk about specifically this problem, but applied to a GWAS context. So check that out if you're interested. And then finally, now you have everything you need to construct a hierarchy. So I'm not going to talk too much about the Clixel algorithm, but that's an algorithm that we have developed several years ago that fit all of the definitions of a hierarchical model that we have put forth in the very beginning. So this model contains about 400 genes, about 117 modules or clusters. I manually remove kind of the clusters that are too small because they generally are just going to clutter up the visualization and they don't generally add too much. So what happened usually then is that once you get to this point, everything kind of just stalls because it is very difficult to visualize a DAG, which is what this is, and because of the complexity of it. And to solve that problem then, we have developed a suite of tools to then visualize and analyze this hierarchical model. So the first step is to visualize. So we have developed a web app called HiView. And what HiView does is that it takes in this hierarchical model and it visualizes it in two ways for you, so you can select which way you like better. So one, we affectionately call this the circle packing, just because you're packing a bunch of circles into a bigger circle. And what this is that you're viewing a hierarchy from top down. So the outer circle is all of the genes in your model. And then the first thing you see, so the immediate smaller circle is then the first layer deep. So the one layer down from the top. So if you don't want to see a top-down model and you want to see everything at once, we produce this thing called the node link diagram. And so we labeled a route for you, which is right here. And what you do, instead of going down, you're going out. So you're going horizontal. So you start here and you want to see the next thing, then you just go out, and that's your next layer down. And then you go out, and that's your next layer down. And so what this produced is that it produced like a fractal-like image where you can actually zoom in and then kind of zoom in into both out and in at the same time. So I want to give you a perspective of what it looks like when you click through it. So let's say we're interested in this particular subsystem. So right now, these terms are just labeled by a random integer. They're not that important, but we'll have tools to fix that. So let's just say we're interested in that particular term. So we're going to click through, and you're going to go in, and what you see is that in this circle packing view, what you have done is that you've gone one layer deep, so now you're seeing the next layer down. So these terms are then the next layer. And so for here, then what you have done is that you have zoomed out. So you have gone out from that central node outwards, and then you start walking out to the peripheral of the model. And then once again, just to see what it looks like again, so you keep going down on a circle packing, you're just continuing to drill down into the model. And then similarly, you're continuing to go out of the model. And so that's one way to browse the model, just to make sure that the hierarchy actually makes sense and it passes some of the properties that you would believe it to have. But then we would also like to analyze the source network, so the network that you use to build this model. So HiView produced this network view where when you click on a module, it returns to you the subgraph of that network and what it actually looks like. And so the weights here, so the heavier the weights, then the thicker the lines. This gives you a proxy for how dense the network is. If you find a big term with not a lot of connections and maybe something has gone wrong in your analysis. But the cool thing about this is that not only can you see the source interactome, you can also start to leverage the publicly available networks. So for example, let's say we want to see how these genes that are highlighted here, let's see how they actually interact in string. So we will select string, and then you can actually see that, well, actually, a lot of these genes are actually supported in string to interact. So that's great. So let's say we want to only look at physical connections, so we can look at BioGrid. And then you do a similar thing, and once again, you have this subnetwork with the evidence supported by BioGrid. And let's say you have a very cool network that you don't want to share with anyone else. You could put it on index, put it on private, but then you can then go ahead and give us the UUID, and we'll visualize it for you. So you can input any custom networks that you have to do a similar analysis. And then finally, I want to call out this Cytoscape button, which if you like this network, let's say you're preparing for a publication, you could easily load this to Cytoscape. As long as you have the Cytoscape app open, this subnetwork will be automatically flushed to your Cytoscape using the CyRes interface. We have also produced one additional tool, and we're calling this the portal for now, and that is to do an enrichment analysis on the gene set based on the vast amount of gold standard networks that Index has already curated. So in this case, it's doing basically an enrichment analysis, and here this particular term is well annotated to the human immune response to tuberculosis. So right off the bat, that could be used as a term name instead of this term like 874, so that's not very informative. This is much more informative. So we could use this service to then go back and annotate these hierarchies. The next thing of interest is that you can see that this term contains two children terms, which are also unnamed. And so I have performed the enrichment analysis, and this is the result. The parent term, so the higher, bigger level term, is actually the human immune response to tuberculosis. And then the children are two specific events, which are the IL-2 signaling event and IL-27 signaling event. After a brief literature review, I was able to confirm that both of these play a role into the immune response in tuberculosis, as well as these are not, this pathway is not specific to tuberculosis, but also occurs in asthma, despite the differences in etiology. This, I think, is a small example to show why it is important to model your networks in a hierarchical way, because not only are you able to do gene sets where you can get these names out of it, but you're also able to understand the interactions of how these smaller terms integrate into a larger term. And when I looked at how IL-2 and IL-27 interact, there is literature evidence to suggest that IL-2 actually inhibits IL-27 in certain contexts. So I think this is just one example of what these hierarchical models can do. So with that, I hope that everyone is convinced and would like to try this out. So if you're interested, there is a GitHub page with a Python package that you can install to then get this pipeline and then try it out yourself. And if you're too lazy to install anything or you don't know Python very well, you can actually hit up our web service, which is in a form of a REST API, where you can just give us a disease ontology ID, and then we'll do the rest for you and give you a HiView link. And so you can just browse. So finally, in conclusion, we have implemented a workflow to automatically build these disease-specific tissue hierarchical models. We have built several tools to then visualize these models, which then acts as a hub to layer additional data. And then finally, Index Search Portal, we think, is a great way to interpret hierarchies, but if you're not doing hierarchies and you just have a gene set that you want to analyze, this is a standalone web app that you can hit separately. With that, I would like to thank everyone who's involved. This is a big effort that involved many people. Many people from the UCSD side have worked on HiView, DDOT, which is the package that underlies the whole thing. ISB people who have contributed ideas to this whole DiseaseScope idea, as well as developing a big gym. And then Jianzhu and then Xiang Wang has also developed tools to help us interpret, which I don't have a lot of time to get into. But so I'd like to thank everyone in the lab, as well as our funding sources, especially for NetBio, for providing funding to get me to this conference. Thanks for listening.
We have time for 1 or 2 quick questions.
Yeah.
Go ahead.
Yeah. Oh, sorry.
Interesting talk. There is a zoo of hierarchical models out there for hierarchical network clustering,
Yes.
and you didn't kind of address what do you do differently, how do you do it differently, are you better than they are? Can you comment on that a little bit?
Yes, it has, yes. So there are a couple methods that I'm aware of. A lot of, so for example, let's say Louvain, that's a very popular community detection algorithm. It does multi-layers in some way. But what that produces is kind of this tree-like structure where you have one resolution, and then that gets integrated into the next level. And we find that that tends to be a very strict way to model kind of these cellular hierarchies. And then the other thing is that, for example, we have also played with something like InfoMap to build these hierarchies. And at least preliminary, some of the problems that we've had with it is that it tends to be very flat and very shallow, and it doesn't capture kind of it's not quite as deep as we would like it to be. So that's why right now we're defaulted to Clixo, but in the Python package, we have actually implemented several other additional methods that if you would like to use, you can.
Last question.
I'm wondering how much HiView handle very densely connected network? Because the one you showed is rather sparse and has kind of a hierarchy into the DAG. So,
yes.
So how does it deal when you have a much more densely? I'm thinking of people doing bait in proteomics when they get a list of 40 interactors for each and every bait they throw in the cells. So can you comment on that?
Yeah, so absolutely. That's a very challenging problem, and that is something that we are working on. So basically, the answer is HiView doesn't really handle that very well, and it basically cuts off as a threshold. If there are too many edges, it's like, yeah, I'm done. But if I remember correctly, what you still can do, though, is then load that network into CytoScape, so you can have a local environment to visualize that. The other thing, though, is that HiView is able to handle large genome scale type hierarchies, though. So the network viewer does have some issues with the edges, but it is able to visualize something like Go, although that's like kind of the upper limit of it.
Great. Thank you very much.
Yeah. Thank you.
Okay, our